Hi everyone. I'm going to talk to you about tape adhesive and IR analysis. My name is Professor Chang Yu and working as a faculty in the Department of Chemistry and Chemical Biology at RPI. So I'm going to talk to you. This is a part of the National Chemistry Day. We'll talk about the sticky stuff and we want to know we can analyze those sticky stuff using technique called IR. So what I'm going to talk to you about is outline, which is what is our IR spectrum result on tape adhesives. So let me show you the result first, and then follow I will explain what this IR spectroscopy means, and then they will show you that how the experiment is being performed in our lab, and then my graduate students are helping me out to finish that. And I will show you the conclusion. So this is we took the pictures. We took some tapes from our home. One is a masking tape, and then this is a different kinds of tape, packing tape. You see the electrical tape, some silver tape. That's a Scotch tape that you see here, and then some other size of tape. This is a duct tape and so on. So what we can do is we can bring this sticky side and putting into the instrument called IR instrument, and we are taking those picture looks like that.
Hi, I'm Keith DeNivo. I'm a fourth year student at Professor Yu's laboratory. Today we're going to measure the different adhesives that's found in common household tape. So you just have to simply take the tape and put it on top of the diamond crystal that's right here. Press it flat. Then you can just say collect sample. These are several different tapes that are commonly found in the household. They usually have different adhesives on there that you're able to measure using the IR.
And this is all the through field samples. Let me show you the first of five samples first. So these are the five samples: the green, gray, packing tape, duct tape, and white tape. They all have this kind of the lines, which is the response from the detector from the instrument and showing as a function of wave numbers. So it is a spectroscopy, it deals with the wave, and they have a different wavelengths of the light or wave numbers. And then when you see that some tapes material shows some response around 1700 wave number, whereas some of them are not. So this means that these three are chemically different from the other two. So as I kind of marked here, number one and number five samples are showing a peak around 1700, and the other three did not show. So if I go to the other samples shown up here, so once again, IR spectrum was being measured as a function of wave numbers, and different wave numbers show the different motions of molecules, and I will explain you in the following slide. And we find 1700, the sample 678, which is silver tape, thick rubber tape, and the electric tape, like electric tape. They have these peaks around the 1700. And whereas also the Scotch tape and the thick white rubber tape, and those are the one that showing this, showing the peak. Whereas this the packing packaging tape, and also this is a masking tape that's cream colored, and then these are the ones that do not show. So they are chemically different from the others. So it's kind of interesting for you to know. This is the main message for today. Tapes are all kind of sticky stuff like this, but they are not chemically the same. Some of the chemicals are the same, some of the chemicals are not, depending on the, I guess, the stickiness they want to control on the surfaces they are being used. So this is a sort of the mini conclusion that these are the tapes that we have looked at it. Those in the white circles are one that's showing some peaks around 1700 in IR spectroscopy. So they are chemically similar. What that means is the one that is not boxed in the red, they are chemically different from those in the box. And I'm going to talk about what is an IR spectroscopy, and I'm going to see some animation showing the molecules in motion. Hey, hi everyone. I'm going to show you some molecular motion using the IR spectroscopy, how they are related. Here's an example of compound called 1-hexene. And there is a peak in the IR, certain wave number around 3000 or even higher. And this is a motion related to carbon and hydrogen vibration in this way. And this carbon is connected by the double bond. And if I go to the other portion, then this is another kinds of motion they call the symmetric stretching. This is an anti-symmetric stretching of the carbon with the three hydrogens. So molecules are essentially at room temperature, so they have a thermal energy to vibrate the bond, and this vibrational motion is captured by this bunch of peaks in the IR. And this peaks around 3000 is particularly useful for us to look at the CH motions. So this is a symmetric stretch. So this is all can be looked at, and they showed in the IR peak. So the molecules are moving. So this is a kind of interesting place where what they call the out of plane motion, when you have a carbon double bond and CH, this is where things are being kind of vibrating, and that's around the 900 peak, and they showed in gray. And this is a carbon-carbon double bond, and that's around the 1600, and this is a carbon-carbon double bond. So this IR spectroscopy therefore allow us to understand the motion of this molecule. This software was provided as a computational model being provided by this PERKIN ELMER IR tutor. And then I want to show you that I talked about the 1700 peak, and that has something to do with the motion in this what's called the ketone. So if you look at here, and this is a carbon and an oxygen, it's a double bond. There is a strong peak here, which is representative of carbonyl peak, CO double bond stretch. And this is what you guys have seen in the adhesive. Some of them have a 1700, some of them has not. And this has something to do with the distinct vibrational spectrum around the carbon ketone.
Hi, I'm Lauren Zarzeski. I'm a second year PhD student at Rensselaer Polytechnic Institute, and today we're going to talk to you about FTIR, or Fourier Transform Infrared. This is our spectrometer right here, and basically this instrument allows you to examine the fingerprint spectrum of any compound or any substance you want. Essentially, you would put your sample right here on the center of this little platform, and the IR beam would come up from underneath and scan into your sample to produce your spectrum on the computer.
So we're going to measure adhesives that are found on common household tape. All you have to do is put it above this sensor here in the middle. And then we can run the IR spectra.
So finally, to a conclusion, what we learned today, we using the different tapes, and we know that not all the tapes are chemically the same. Some of them are same, some of them are different. And we have used an IR spectroscopy to identify tapes of this kind, such as this green masking tape, and this white tape, and the silver tape, and also the scotch tape and those electric tape. They are the similar kind, but those with the packing tape and so on, they are chemically different. And how do you figure it out? We're using the vibrational spectroscopy. I have shown you some vibrational motion of the chemical bond, particularly the carbon oxygen double bond vibrates at the 1700 per centimeter wave number, and then some of them has a ketone group, CO double bond, some of them do not. So we learned that tapes can be analyzed using IR spectroscopy, and we find that some of them are same, some of them are different. Thank you for your attention, and it was nice meeting you.